everybody. For today's journal club, I'm going to be presenting on Crumblr, the paper titled Fast Flexible Analysis of Differences in Cellular Composition with Crumblr. And this is a paper by Gabriel Hoffman and Panos Roussos. And it's a preprint. I guess pre-printed this January. So hot off the press. So they open the paper by discussing issues about like modeling with cell fractions. So basically, if you wanted to say in cases there's more of cell type A than in controls, an issue is that if you like, I guess, summarize your cell type fractions as a proportion. So for instance, if like 1,000 of 5,000 cells are cell type A, you reduce that to like one, you lose important information about precision. And this is what happens in a couple of transformations of the data, such as center log ratio transformation or binomial modeling. But then on the other side, if you have models that consider precision, these are not great and have high false positive rates. So there's the Poisson negative binomial models, as well as the method scCODA. So this is kind of like what is out there and like what the issue was. And they propose a solution by introducing their new model, count ratio uncertainty modeling, based linear regression, or Crumblr. So it's a fun little acronym. Imagine like the crumbs are pieces of the whole, so like cells are piece of the whole tissue. So basically this method helps us perform differential cellular composition with modeling variation in precision. And it models the fractions considering precision while for controlling for that false positive rate. So kind of like the best of both worlds from the previous models. And so their method is to transform cell counts with center log ratio. This is kind of similar to ILR, which was a method that we used to like compare and contrast cell fractions previously. And they use the Dirichlet multinomial distribution estimates to account for sampling variance. And with these two, I guess, models, we're able to do univariate. So univariate, where we test like just one cell type, and also multivariate testing. And then in this paper, they show the application of this, and they apply Crumblr to 4 single cell data sets and like test for cell type differences across some different variables. So figure 1, they show their analysis workflow. They show that Crumblr takes the count matrix and also the study design and these variance partition to basically show like what variables have big impacts, explain the most variance in the cell fractions. And then they also do the regression testing for differential frequencies. And then you can use some different, and this is, I guess, you can use different methods here, like Dream, LMFIT, or eBay's. And then they also show the multivariate testing, where they use MD test and tree test, where you can test for differences at like, I guess, D here helps explain that, where you can either test for differences across like case control or your variables at a cell type or at the, I guess, like parent of the cell type, so at a broader cell type. So for instance, you could cite like all your excitatory neurons, or excitatory neuron 1, excitatory neuron 2, or like excitatory neurons as a class. So they have that multivariate and univariate test, which is cool. Also in this figure, they show how the Dirichlet multinomial helps us estimate the proportion versus standard deviation. And they kind of demo the modeling of Crumblr versus the true and the CLR differences. In 2, they did some simulated data to show how Crumblr overcomes or like performs and also overcomes the false positive ratio. So they're kind of demoing that model that include precision often have power. So in 1, it's area under the curve. So that's kind of showing like the performance of the model. area under power regression curve. So these are all pretty comparable. You see Crumblr is high. We also see binomial and Poisson as like having high area under the power curve. But if you look at the false positive rate, you see the Poisson and binomial stand out. So Crumblr kind of being one of these tops that then has a low false positive rate. And then you can see the area under the curve, under power curve, kind of increasing with the number of samples and number of clusters tested. You can see that this is outperforming, and these are the Lin-Sullivan methods, outperforming the Fishers and Seduk method. So they conclude that this empirical Lin-Sullivan method has like the high precision, like high precision recall, but then it is also going to have that low false positive rate. Or I guess, so the green and purple here match up like almost completely. Then if you look at false positive, the green, so the empirical and Sullivan's is the lower of the two. Moving on, the following figures kind of like show or demo the utility of this and like how you can actually apply these to like real data sets. So in first one, we're looking at changes in age, changes in cell fractions associated with aging in blood. So we have lots of single cell samples out of blood, and you know, they're made up of different cell types, and we have those fractions for all of our patients. So for A, we can look at variance partition, and we can, so variance partition again, it shows the percent variance explained by this percent of the variance explained for a cell type's composition, and how much these different variables contribute to that. So pool number, age, and sex are all the ones tested, and the residuals are kind of like the variance that we can't explain with the tested variables. So we can see in the chart, we see lots of red. So a lot of the variance is explained by pool number, which has to do with like the actual like batching. So that's kind of technical effect. For age, which is more like the biological question, we see that that has a large effect size in a few of these. So namely native CB8 plus alpha beta T cell has a large blue bar. So we know that that's like probably age explains a lot of that variance. There's a couple others that have a higher age percentage. We can also see that sex accounts for some of the variance in these cell types, but age in this cell type is what we're going to focus in on. So in the second panel, they're exploring effect size tested by age, and basically either positive or negative relative to age. So we can see that this native CV8 plus cell type has the largest effect size and has a negative effect with age. If we look at C, this is then doing like the univariate or multivariate testing, where we can see that like this univariate test has like a significant negative effect size, so it gets blue and a plus. And we can see that on this like branch of the dendrogram, right, that these other ones are also negatively affected and significantly so. So these are like related cell types that have a similar effect. So this is like an interesting conclusion you can draw from that. And also down here, you can see like on the flip side, the branch of CD16 plus monocyte cells and related cell types seems to be like affected positively as a group over age. And then D is kind of zooming in on the data itself. So we have age versus the CLR value for the native CD8 plus cells. And you can see that this is like the modeling over that. So they have the quadratic fit with and without the Crumblr metric precisions, either the green or the purple. And another thing that they account for and show in these plots is the error. So things with a lot of cells, we can say that like the fraction is like a lower error. So like for instance, down here, 0 out of 855 cells, like that's like something that's going to have like a higher error. So it's red. So you can see that we're considering the precision of these measurements as well as the actual values. So yeah, another similar one. Here we're testing for composition changes with tuberculosis in T cells. So again, A being at variance partition, what are the like what is contributing to the differences. Here we're evaluating more metrics. We see that there's a large contribution from red in a lot of these. So age being like a very high contributing factor. I guess that what we're kind of interested in is green, which would be TD status. We see that that makes a smaller variance. That makes up smaller variance in a lot of these. We do see it stand out for CD4 plus here. Looking at the effect size, then testing for AIDS or tuberculosis, because that's the variable we're interested in, we see this has like a negative effect here. And over here, we see some positive effects in some of these cell types. Again, looking at the hierarchical clustering, we can see that these like negative ones kind of cluster together. We can see that the parent node of these different CD4 plus cells seem to like all have like a significant decrease with tuberculosis status. Likewise, down here, we can see some smaller effect sizes, but some of these are still significant for these some of these some of these cell types down here. So all have very long names. And then D, we can see how we can show the same modeling that we did last time, but instead of a continuous, we have a categorical vector, a categorical variable of case control. And again, color showing that error. So like these red ones being some of these like higher error cell types or measurements. And then 5, again, kind of the same story. So we're getting good at reading these plots. So we're looking at composition change of metastases from prostate cancer. So again, we have our cell types. We're looking at the variance. We can see that the variance partition is really highly explained by patient, which is red, and then also blue, which is disease status for most of these. And then in a couple of the cell types, you can see patient status. So we can actually explain a large amount of the variability for most of these cell types. It's kind of a contrast from some of the last ones we saw. And then in effect size, we're looking at either solid or involved, which are two different tumor statuses or tumor types. And we can see that the effect size for the parasites, you can see we have really high positive effect size for parasites in its related cell types. We can see that node positively increase. I think with it being solid, the solid versus involved. And then on the flip side, we can see that the erythroid cell type, negative effect size here. So sometimes you can see that the groups kind of move together. And then likewise, again, we can show our CLR estimates and the error estimates across these 4 different categories of disease status. All right, figure 6, I believe this is the last example. Yep. And so we're looking at identified changing from infection. So this is cell type changes in the lungs with or without COVID. Again, exploring these variables, we see disease state makes up a large percentage of the variance in these cell types. Looking at the effect size of severe COVID versus healthy, we can see some big changes pop out at us. These MAI T cells, plasmacyte dendritic, looks like invariant NTK cells all have negative effects. And likewise, we have a couple strong positive effect sizes. Here, looking at the dendrogram, we don't see kind of like a lot of nodes that like change in the same direction. So that can be different across different tests or cell types or tissues. So this is like a cool way to break that down. And then again, so where we have the CL values versus our different COVID statuses. So yeah, looking at the monocytes, we can see that they increase across these different categories. And then another way that they presented that is if you wanted to explore a lot of cell types, they made this heat map of sorts where it kind of represents this, but like broken down. I think it shows the effect size across. You can kind of like look and see if it's like becoming more positive or becoming more negative. So this is another way to display the effect size that we see in B. All right, that was it. So they have that same like kind of like range of plots. I think this kind of summary plot would be really useful for a lot of our data sets when we're making in our single nucleus data as we're moving into single nucleus in case control and other disease status experiment designs. Yeah, so in summary for this paper, it's a tools for testing different cellular composition. It considers precision and controls that false positive rate. Within the package, they have tools for variance partition, univariate, and multivariate testing over continuous or oracle variables. And it integrates with single cell experiment and a lot of other tools we're familiar with from Bioconductor. And I think this will be really useful for us at Liber, like I said, as we move into case control single nucleus RNA-seq data sets. Cool, that's all I had.